Welcome to Complex Trait and Evolution session. The first talk is by Yoon Ding.
So I got this talk twice by accident, so I can't cancel it, so apology here. But hopefully you haven't heard about it before. And also, I'm going to tell you something like new and also a little bit crazy at the end. So hopefully that can help a little bit. I'm going to start with a very well-known quote. This is from last night. So I think this is the most simple way to explain like my obsession in evolution. So particularly, I'm interested in behavior evolution. So I think animal behavior is just awesome, and there were like amazing diversities between different animals. So the major question I'm trying to understand is, what's the genetic basis of behavior evolution? Particularly, which genes, what kind of genes and mutations are often used by nature to give new behavior pattern? To approach this question, I'm using Drosophila courtship sound as my model system. So here is Drosophila melanogaster. The bigger one is a female, and the small one is a male. So the male is trying to impress a female. Not very musical, but at least it tried. So there are two components in the sound. So there's pulse signal and sign signal. And this signal can be well quantified. And also these signals show enormous amount of variation among Drosophila species. So here's my favorite species, Persimmilus. Almost like a frog. And also, a nice thing about this system is that some species can be crossed to each other, so this makes genetic mapping possible. The story I'm going to tell you today focuses on a courtship sound difference between two closely related Drosophila species, that's Drosophila simulans and Drosophila melanogaster, and Drosophila mauritiana. Like melanogaster, both species sing pulse and sign sound, but differ a lot in many sound features. So particularly, the fundamental frequency of sign sound, which you can understand as the pitch or the tone of the sign sound, is different. So here, I'm picking wild-type isolate from each species. So it's SIG5 from simulans and Mauritiana and MAU29 from Mauritiana. I'm going to play their sound for you. So first is SIG5 and MAU29. I hope it's clear that MAU29 sings sign sound at a higher pitch. So we want to ask what gene and mutation is giving this difference. So, but if we really want to identify the causal mutation, we're literally talking about finding one mutation among 4 million possibilities. So how do we pick it out? We start with QTL mapping using backwards population. So here, color represents the species identity of DNA. White is simulans, and blue is MAU29. And for the rest of the talk, I'm going to use the same color representation for the species. Here is a QTL map using SIG5 backwards population. On the x-axis, you have the genomic location. Y-axis reflects how likely this loci is linked to a trait variation. So we can notice there's a single QTL peak on the third chromosome, and QTL mapping using Mauritiana MAU29 backwards give a very similar pattern. So this indicates the sound difference you just heard is probably attributed to a single locus on third chromosome. So we further conform and narrow down this QTL using introgression mapping, where we introgress a small piece of DNA from one species to the background of the other species. We ask, does this piece of DNA come from trait variation? But to facilitate introgression of DNA in the QTL region, we develop a collection of fluorescence eye marker lines. So here is how a yellow fluorescence marker looks like. So we can choose one that is inserted near the KLTR region, and for each bycross generation, we can follow this marker, so this can make sure the introgression region is preferentially enriched in the region of interest. So here you're looking at several introgression lines that is generated using this strategy. So the first three show the sign-sound difference, while the last three don't. So this helps us to narrow down the causal region to 140 kb. So it's not 140 kb is not bad, but still too big to find the causal gene mutation. So we developed a targeted mapping strategy. Using CRISPR technology, we insert red fluorescence marker at specific sites flanking this interval, so we can easily identify the recombination events within this small interval just by looking at eye color. So here, all the offsprings without this marker should have experienced recombination that we want. You're looking at the two recombinant lines that define the minimal interval. So the first line shows the sign-sound frequency difference, while the second one doesn't. And their breakpoints only differ in less than 1 kb. And this 1 kb is located within a single gene called slowpoke. So who is slowpoke? It got named from this Pokemon character because the mutants like slowpoke is just very slow. So this is because this gene encodes a calcium-activated potassium channel. During action potential, this gene helps the export of potassium and contribute to repolarization phase of the memory potential. So very typical electrophysiological defect of slowpoke mutant is this prolonged repolarization phase. So slowpoke is a good candidate gene, but we're not sure if it's a causal gene because genetic mapping can give you is always the DNA region, but you don't know if the regulatory sequence of another gene like far away. So to prove that slowpoke is a causal gene, we use reciprocal hemizygosity test. So this is how it works. We can make slowpoke loss of function mutant in SIM5 and cross to the wild-type species of MAL29. And we can do it reciprocally. So we can compare the slowpoke allele from both species at identical hybrid background. So here, the left genotype carries a single functional slowpoke copy from SIM5, and the right genotype carries a single functional slowpoke copy from MAL29. So you can notice that MAL29 shows a higher sign-sound frequency than SIM5 allele. So we can conclude that slowpoke is a causal gene. But interestingly, all the other courtship sound phenotypes, like the pulse sound frequency or the interpulse interval, and the flight wind beat frequency are the same between the two genotypes. So this means that the evolution of slowpoke really changes a very specific aspect of behavior. So this is quite remarkable considering how pleiotropic slowpoke is. It is this essential ion channel gene. It's extremely widely expressed, and it affects perhaps all locomotor behavior. If the fly lose slowpoke, the males can barely sing any signs of pulse sound. So obvious question is, what kind of mutation has caused such a specific phenotype of such a highly pleiotropic gene? So we compare the sequence between SIM5 and MAL29 in the 1 kb minimal mapping interval. There are about 10 candidate mutations in the interval. 2 of them are in the exons, and both are synapse mutations, so they don't change protein sequence. The rest of them are in the introns, including a 6.7 kb retro element. So when we sequence this region in multiple simulants of Mauritiana lines, we actually found that these sites are generally very polymorphic. So making it possible to use reciprocal Hamilton-Gaussian test to further narrow down the causal mutation. So long story short, finally, we narrowed the causal mutation to a single candidate, which is this retro element insertion specific to SIM5. As expected, when we remove this retro element from SIM5 using targeted deletion, it reverts the courtship sound phenotype. So this really proves that this retro element insertion is a causal mutation. But I do want to remind you that even when we do see a general courtship sign sound frequency difference between simulant species and Mauritiana species, this retro element insertion is not a fixed difference between the two species. Actually, it represents a rare variant arising within simulants population. Further, we found that it is a retrovirus, but it's different from all the non-retrovirus lineage. So I get to name it, and I name it Shouder. It's another Pokemon character. So Shouder is this toxic and a little bit grumpy-looking little thing that bites Slowpoke. But when the bite happens, they can co-evolve into a more adaptive form. And depending on where the bite is, Slowpoke can be either more active or more intelligent. But I do want to cut it off now, before this is going to be the only thing you're going to remember from this talk. I want to summarize my story because this is the real story. So using unbiased genetic approach, we start from a natural courtship sound variation and map the causal region to less than 1 kb. So this allows us to identify this intronic retrovirus has modified the function of an ion channel gene and makes the fly sing differently. So I can try to make some general implications from this story, but really, a general understanding of how behavior evolves will require many examples like this. But as we know, the genetic mapping of behavior traits has been notoriously difficult. So major reason for that is behavior traits are often very sensitive and variable. Take courtship sound, for example. Even though they're among the most quantifiable traits, I still have many days that fly just won't sing. And also, sometimes I have so many variations in my data, I just can't make real sense out of it. This has been a real obstacle in genetic mapping of behavior traits, especially for cases with loci with smaller effect. And the simple fact is, if you can't detect the behavior, the phenotypic difference with a reasonable number of sample size, you just can't map it. So in the next few minutes, I'm going to request you to think a very different problem. So the problem is, how can we make the fly behave consistently and robustly? So we use optogenetics. So optogenetics is a way to induce certain behavior by activating the related neurons. I'm going to scale all the technical details of how we made it work, but I want to show you that we can use this technique to induce courtship sound in non-monogamous Drosophila species. So here is Drosophila yacuba. There's only a male in the chamber, so there's no female around. The quality of the video is a little bit shitty because some technical difficulties, but I'm going to play for you. So this is what happens when I use optogenetics technology to induce the pulse sound in Drosophila yacuba. And next will be the similar experiment I do in Saint-Omer. So So I hope you can notice that centromere flies sing a lot faster than Yakuba. This is because the induced pulse sound actually recapitulate the natural courtship sound really well. And the speed difference you just heard actually represents the interpulse interval difference between these two species. And also, a nice thing about this system is that you can see there's very little variation in the interpulse interval. So this is great for genetic mapping. And another amazing thing is that I can make them sing like this forever. I can literally make them sing like 20 minutes, but only have 15 minutes for this talk. So I'm very excited to try to do the genetic mapping on this singing robot. So as a summary, I've taught you our two major efforts in genetic mapping of behavior traits. The first is the genetic strategy that allow us to increase the mapping resolution. The second is the optogenetic strategy that make us to be able to quantify behaviors better. So the genetic basis of behavior variation has been this long-standing hard problem, and I'm hoping that marrying the two efforts can make the behavior traits more mappable. I just want to thank all the amazing colleagues I feel so lucky to work with. Augusto Barroca was the undergraduate student in the lab that did amazing work in the QTL mapping. And a lot of the genetic reagents for the optogenetics tools comes from a joint effort between me and my wonderful colleagues, Jessica Candy. And I also want to thank my boss, David Stern, for all the supports, and HHMI for funding us.